We're now going to discuss glycerophospholipids. You'll notice with glycerophospholipids, phospho is in the middle of it, meaning phosphorus. It's referring to this phosphorus down here. This is very similar to the lipids that we referred to before with the glycerol and the 3 fatty acids. But instead of the third fatty acid, we have an amino alcohol down there instead. This is making an area that's extremely polar because of this positive charge, which means it's going to interact very regularly with water. Here's a couple examples of some phospholipids. Notice that they still have the carboxylic acids here, the fatty acids, and that area is nonpolar because of how many carbons and hydrogens there are. So therefore, it's not going to want to interact with water. However, because of this positive charge here, that's going to want to interact with water because of the opposite charges. Oxygen's partially negative, hydrogen's partially positive. If we look at a difference between trialkyl glycerols and glycerophospholipids, they both have the esters in the first two. But the bottom one has the ester, while the phospholipid has the phosphorus. And that's the only difference, and that difference makes a huge difference in the way it interacts in aqueous environments. So they have a polar and a nonpolar region. The polar is referred to as the polar head, and that's the thing that contains the amino alcohol and the phosphate. It is strongly attracted to water, which is really important for biological reasons. They also have a hydrocarbon tail, which is making that nonpolar, just like the other lipids. It's the most abundant lipid in cell membranes because cell membranes are made up of these. They also play an important role in other aspects too, like the delivery of compounds from one location to another. So once again, here's the polar head, the one with the charge on it. That means that's going to interact with water. And these are nonpolar tails, which do not interact with water. So we have an area, we have two distinct areas here. One that will act with water and one that won't. And because of that, we're able to create cell membranes. And our cells are then able to flow within our bloodstream. An example of that are lipoproteins. They transport lipids. Lipids are nonpolar, so therefore they're not going to mix together with the water very well. So what happens here is we take the phospholipids. The green heads here are those polar heads. These polar heads are then going to interact with the water or the aqueous solution. In the interior, these are all nonpolar compounds in the middle. That's what's being transported. These nonpolar tails are making it so that the compounds in the middle can interact with the nonpolar tails. If it was just a completely polar area, so that way it would react with water, it wouldn't interact with the stuff inside, and so you're going to have the problem inside of it. So by having both, one that will interact with water and one that will interact with nonpolar compounds, you're getting the best of both worlds. So this whole thing is then going to be able to dissolve in our aqueous biological stuff like blood and transport it from one location to another. We're going to discuss this more when we start talking about cell membranes.